Hello everyone, my name is Chinni Jyoti Prakash and I am a final year undergraduate pursuing my bachelor's in computer science and engineering from IIIT Vadodara International Campus D. Today I am going to demonstrate my work on analysis of gene casualty using embeddings. So before going into that, I want to discuss some basic terms related to it. So we'll be having phenotypes, genes and everything. So phenotypes are nothing but the characteristics or traits which we can observe in a person, and genes are the responsible for that. Genes are inherited from the generations, so it causes changes in those characteristics. So, and another term is there known as causal genes. So causal genes directly influences the phenotype. So any minor change in the causal gene could completely alter the phenotype. So finding that casual gene would help in diagnosing and many other treatments. So, in this work, I'm going to explore the relation between the phenotypes and genes embeddings, which can give us insights about the gene casualty. Let me discuss the problem statement now. So, in this problem, we are given a data set with phenotype embeddings and gene embeddings. So, embeddings is nothing but the mathematical or vector representation of these phenotypes or genes. So our goal is to determine how these embeddings would tell us the causal gene. Now, by applying various machine learning algorithms, we'll find out it. So our primary objective is to map those phenotype embeddings to causal and non-causal genes, and we'll perform dimensionality reduction, and we'll perform clustering, and finally we'll find out the similarity, and we'll provide the insights about the data. Also, let me discuss about the data set. So in this data set, we'll have four types of files. First one is phenotype embeddings, which are the embeddings of the phenotype and gene embeddings, and phenotype gene mapping. So it is the relation between the phenotypes and genes. And the last one is the causal gene labels. It contains whether the gene is causal or not. So this is about the data set. Now let me discuss the workflow. So we can see this through this chart. So firstly, we'll load the data and perform some pre-processing methods. And after that, we'll perform to perform dimensionality reduction, we'll use principal component analysis. And after that, to perform clustering, we have to find out the perfect k value. So we'll perform elbow method, and after that we'll perform clustering, and to find out the similarities we'll use cosine similarity, and finally we'll evaluate the similarities, and I will get the insights about the casualty gene. So this complete data about each and every process we'll discuss in the code. So let me dive into the code. Now let me get into the code part. So this is the Python notebook file. So this is the basic details about the title and the description. So actual code starts from here. So we'll be extracting, so we'll be downloading, extracting the files from the Zenodo URL. So using wget, will first download the file and we'll extract here, and we'll first install and import the necessary libraries, like for manipulation, data manipulation, we'll use pandas, and to perform clustering and scaling, we'll use scikit-learn, and to find the optimal k value using the elbow method, we'll need for a need. So, and these are generally for data visualization, we'll use matplot and seaborn, and here we'll perform the dataset loading. So we, as I said before, there will be four files: phenotype embeddings, genotype embeddings, phenotype mappings, and casual labels. So we'll load those files into these, and turn them into the data frames, pandas data frames. As mentioned in the task, we'll generate a random seed based on hashing my name. So here we're taking my name Chinni Jyoti Prakash, and we'll using SHA-256 hashing algorithm, and we'll get a seed value, and we'll be taking 500 samples as to create subsets of data. So this is one of the sample phenotypes data. And when it comes to pre-processing, First, we'll remove the unnamed or zero values. Uh, so basic pre-processing will be done, and we'll be converting that embedding strings into lists, and we'll perform parsing on it. So like to convert those strings into actual floating numbers, we'll perform parsing, and those here those parsed embeddings are went into further processing here, and a sample genotype is taken. Sorry, the sample phenotype embeddings are taken, and those parsed embeddings are converted into data frames. This is all about the pre-processing and cleaning. As we can see here, we are performing parsing, and we are converting the parsed embeddings into data frames, and we will taking the sample here. Now, yes, now we'll be discussing the dimensionality reduction using principal component analysis. So before performing principal component analysis, we have to standardize the embedding. So standarding means, the we have to scale the embedding so that their mean value should be 0 and the standard deviation should be 1. So for that, we'll be using standard scalar, and we'll be scaling both gene embeddings and phenotype embeddings. And after that, we'll be performing principal component analysis on that embeddings. Um, the number of components will be 2. We'll be reducing the dimensions to 2 to get better results about visualization. And next, here we are using elbow method to compute optimal number of clusters. So here in it is elbow method is like hit and trail method. We'll start try using different k values from 1 to n. So, and we'll plot it in a graph, and whenever there is no changes, we'll fix it as an elbow point. So this is what happens in the elbow method. Here we are taking within cluster sum of squares, and we'll using C for k in k. We are using a for loop for trying each and every k, and we'll be fitting that model. And after we'll be plotting the elbow curve. So this is for the phenotype embeddings. We'll perform the same with the genotypes. So here we can see the plot. So elbow method for phenotype embeddings. So we can see here there is not much change after the number of clusters k value 3. So we can assume that the optimal elbow point is 3. The same actually happened with the gene embedding. So we have to make the data into 3 clusters. Here we are performing the clustering with k-means optimal with k-means algorithm. So here we are taking the k-means and plotting, for both gene clusters and phenotype clusters, we are fitting the k-means using those gene embeddings and phenotype embeddings. Here are the plots of genotypes, embeddings, and phenotypes. Okay, this is the gene type, gene embeddings with k-means clusters 3. So we can three types of clusters there, and with also phenotype embeddings, we can see there are 3 types of clusters we have used. Next, to find the similarities and the relation between the phenotype embeddings and gene embeddings, we'll perform cosine similarity matrix calculation. So it is a part of vector analysis. So cosine similarity matrix, it gives the how much the phenotype embeddings and the gene embeddings are related. So so here it takes cosine similarity matrix. Will be taking it and perform cosine similarity. So it gives the values between 0 to 1 as, if there is no relation between the embeddings, gene embeddings, and the phenotype embeddings, the cosine matrix similarity value will be 0. If they are perfectly aligned, so then the value will be 1. So there won't be any perfectly aligned uh, genotypes and phenotypes. So the values may range from 0 to 1, but not exactly 1. Here we are taking, so to visualize it, so we are taking some samples. So the rows contain the phenotypes and the columns contain the genes. So this is one of the samples. So here we have a phenotype called Parkinson's disease. ATM-IN genes. So there is 0.27% similarity between those phenotype and genes. So similarity matrix just tells us how strongly they are related. And finally, to give us some insights, I have calculated. So the top five values with most similar values between the phenotype and gene pairs. So, and I also calculated the least similar phenotype and genotype pairs. So here are these top five similar phenotype pairs and top five similar least similar phenotype gene pairs. That's it from the code. So finally, these are the results we have obtained after performing k-means clustering. So this is for the gene embeddings, and this is for phenotype embeddings. So to show you the cosine similarity matrix, I have taken some random samples. So this is the cosine similarity matrix, which contains phenotypes in the rows and genes in the columns. And these are the top five phenotypes with most similar phenotype and gene pairs. So this is all about the analysis. So thank you.